The University of Michigan Division of Gastroenterology and the Inflammatory Bowel Disease Program host the IBD Visiting Professor Lecture Series in Ann Arbor, Michigan. This series presents the latest in clinical research and basic science from IBD experts from around the world.
Warm thank you for inviting me here. It's both an honor and a pleasure to be here at Ann Arbor. It's my first time, but the impression is overwhelming, to tell you, just by meeting the people and also the university. It's quite a setup you have here. I did change my title a little bit, so I cheated on Peter, because if you look at the flyers, it says like short-term outcomes in IBD, and I don't think they are relevant. I think we always should look long-term for all our patients. So I'm going to make short-term and long-term a little bit, and I'll make it clear to you. Just as a sort of introduction, you probably don't know that, but a lot of the people from the region where I live moved after the First World War to this region, Michigan, and specifically to Detroit. Our country was in chatters after the First World War, and so there is still a Dutch-speaking Gazette from Detroit that is published since 1914, and so there's quite a few people from our region that moved here are still here. And just evidence-based medicine, I Googled my name and Detroit. And sure enough, there's a Joseph van Assche, the third, so probably his third generation. There's some plumbers, so it runs in the family, fitting tubes. There is a Van Assche construction not too far away from here. And Linda sneaked into the animal hospital. She changed her name to stay under the radar, but I still found her. So there's only my last name, and it's not a very popular last name in Belgium. It's all over Michigan. These are my levels of contamination at this time. So I don't have to repeat this to you, but you all know that IBD we don't really know what's going on, but by any means it's an interplay between immune factors, genetic factors, environmental factors. And what is happening in a patient is this vicious circle of inflammation, tissue damage, loss of organ function, and surgery. And this translates for the patient in symptoms such as pain, diarrhea, and a lot of other symptoms. So let's look at those symptoms a little bit more closely. Now, to bring you down on earth, this would be the ideal drug for IBD. 100% maintenance remission, induction of remission, and 100% maintenance, no loss of response. Well, keep on dreaming. We don't have that drug. These are steroids, too toxic, so we all taper them down. Azathioprine, 6-MP, you can have a steroid-sparing effect. And to be honest, with the anti-TNFs, we're probably in the same ballpark at this time. So there's this gap, therapeutic gap, that we don't fill for our patients. So my question to you is the gap so wide, or are the endpoint of the clinical trials maybe not perfect, and also the selection criteria? And that's what I'm going to talk to you about in the next 10, 15 slides. This is the reality of anti-TNF agents, like the new kids on the block, where everybody talks about. Well, if you go across the board of all the agents that have been tested, both in UC and Crohn's disease, you see anywhere between a 20% to 40% non-responder rate at onset. So that's the reality. Are these all non-responders? We don't know. And again, to add the level of complexity, there is a difference between the patient you see in your office and a patient in a clinical trial. You all know that, but I just wanted to point out the differences again for you. In a clinical trial, there is a strict patient selection, both for avoiding adverse events and increasing efficacy. You know that. You do some patient selection to a certain extent, but you can't do the same patient selection as is being done in clinical trials. Your patient selection in Europe and Canada is based on label restrictions. I know that's a little bit less in the US. And cost issues, right? Prospectively defined efficacy endpoints are very important in clinical trials, but it's not what we're using in clinical practice. We use more of a physician or a patient global assessment. And then, of course, the CROs are there to have a very strict monitoring of your trial conduct. And they always say, like, the FDA is going to audit you, so please, doctor, comply. Please, nurses, coordinators, comply. Well, What we do is our routine follow-up, and I'm sure the U of Michigan follow-up is different from that in the Mayo or Toronto or the one in Leuven. So we all have our tricks to the trade that are not in a protocol. And of course, a study is double-blind, or should be double-blind, where we're all employing the placebo effect. Whether we want it or not, we're doing that. And of course, trials are designed to show efficacy. While you're a problem solver, you don't have any priori design for your patient. I mean, you try to tailor all therapies to your patient, but as post hoc, you're solving problems. And this is a sobering slide, but it's very true. I mean, the Crohn's disease activity index that is used by EMA, by FDA to judge trials and that is employed by companies has a lot of subjective component. And you know that. Severity of abdominal pain, number of loose stools, general well-being, it's all quite subjective. You see, no better. Number of stools, rectal bleeding, probably rectal bleeding is a little bit more objective, but still it's reported by the patient. And a physician global assessment, what's that? Where's the wind coming from today, right? So it should be objective, but we all know there's biases in there. And it's no surprise that if you give, if you give IBS patients the CDAI, they may have mild to moderate Crohn's disease if you just don't think of them as patients with irritable bowel syndrome. And this patient, for example, they will have pain, but what's the cause of the pain? Sure, there's ulcerations. You can see the cobblestoning. But is this the cause of the pain? You don't know. It could be obstructive symptoms also due to the fibrosis. So it's really in the air there what your patient is suffering from. And the ancient books of the medieval physicians and surgeons had for inflammation the characteristics of rubor, tumor, dolor, et calor. And so the dolor is the pain. And where does it come from? Well, we know that active inflammation triggers afferent nerve impulses. And a lot of the motility people in this room write article after article about post-inflammatory hypersensitivity. And that's probably also occurring in patients with IBD. But also strictures may provoke obstructive pain due to a pre-stenotic increase in wall tension. And that's not necessarily linked to inflammation. We have to be careful. So this is just a panel of the studies that have been reported since 2004 or 2002 on patients with clinical remission IBD having IBS-type symptoms. And you see across the board, for UC, there's a 32 to 40% prevalence of IBS-type symptoms, Rome 1 or Rome 2 criteria. Crohn's disease even higher. The last report from the group in Cork is 60% using the Rome 2 criteria. So yes, we're dealing with symptoms. Patients in remission on the CDAI, they're still having symptoms that look like IBS. I'll come back to that at the end of my presentation. Is this IBS or is it something else? It's surely not IBS. We shouldn't call it IBS, but are these functional symptoms due to motility or hypersensitivity, or is something else going on? And this we should not forget. The placebo effect is there. And here I'll switch over from symptoms to biomarkers. Because what we've learned from the biomarkers is that what they do is bringing down the placebo effect, if anything. First example, open label experience, Liège in Belgium. Edouard Louis was the first author. Already before this thing came about, he was looking in 2002 at his patients treated with infliximab, only to show that in patients with an increased CRP above 5 milligrams per liter, there was a much higher response rate than in those with a normal CRP. So first indication. Do we have clinical trial data? Yes, we do. Certolizumab pegol, one of their first studies reported was negative. But what was happening, if you increase the CRP at baseline, so you just have stratified post hoc for the patients with an increased CRP, what you saw is that the placebo rate was going down with increasing CRP, not so much the active drug rate.